Hi and welcome. I'm trying to learn cytology. Today, facilitated diffusion, and after we've done that, we're going to do extensor carpi ulnaris strength training. And if you participate in the extensor carpi ulnaris strength training, then it's already on this. Facilitated diffusion. Several solutes that are too polar or highly charged to diffuse through the lipid bilayer and too thick to diffuse through membrane channels can cross the plasma membrane by facilitated diffusion. So they are too polar. They cannot just slip through it like water just through the wall out of the phospholipids and glycolipids. And they are also too big to fit through the channels, as potassium can do. It was an example, potassium ions. Now they are bigger and they need facilitated diffusion. This process originally was envisioned as one in which it was understood as one in which a carrier protein bound to a solid and diffused with it across the membrane. That makes a lot of sense. But they say that's not the case. The name is unfortunate, however, because the process is not truly diffusion. I don't really know what diffusion means. I think that means to go and become a part of the other substance. The name is unfortunate, however, because the process is not truly diffusion. Instead, the solid binds to a specific transporter on one side of the membrane and is released on the other side after the transporter undergoes a conformational change. Vis-a-vis transporters here, transporter channels. But it makes sense. The channels are specifically for one substance that it can go just through it. They said 1 million potassium per second. More than 1 million potassium ions can flow through a potassium ion channel in 1 second. And the transporter can take bigger ones, but it need longer. Okay, through conformational change. I keep on calling them snakes. Like snakes taking it in and bring it in. For this reason, because they're conformational change to it. For this reason, some researchers advocate changing the name to facilitated transport. What is the difference between them and transporters? Maybe because it has something to do with active transport? Okay, we go on. It is not diffusion, it is transport. It is maybe the transport thing, it is maybe done by transport, and it is or is not active transport. Maybe or maybe not. Like diffusion, nevertheless, the net result is movement down a concentration gradient from a region of higher concentration to a region of lower concentration. So if there's more of that one substance, it goes from there to a place where there's less of a substance. This happens because the solid binds more often to the transporter on the side of the membrane that is more concentrated before being moved to the side that is less concentrated. How does it know it should let it go off instead of binding it again and transporting it again over? And before letting it off, binding it again and transporting it back. Does it sometimes get happening? It just transporter is transporting it the whole time from one side to the other? No, not that, but something else happens because the solute binds more often to the transporter on the side of the membrane where it is more concentrated before being moved to the side that is less concentrated. Once the concentration is the same on both sides of the membrane, solute molecules bind to the transporter on the cytosolic side and move out to the extracellular fluid as rapidly as they bind to the transporter on the extracellular side and move into the cytosol. How do they know that they should not go directly back? Because they just release out, and if they go around there, maybe they come back and then they go back. How does the transporter know that it should let now off and not bring it out again? And by your next one, you should take it. Thus, the rate of facilitated diffusion is determined by the steepness of a concentration gradient between the two sides of the membrane. So it's just a transporter bridge going from one side to the other one, always picking people up, or these whatever molecules, bringing them over all the time. And if in the morning all the people want to come from here to over here, then there are here 1,000 people and here just 5 people wanting to go over, so it drives over with 100 people and takes just 5 minutes, and 100 people and 5 people. In the afternoon they all want to come back, and now it's 100 people from here and just 5 people back. The number of facilitated diffusion transporters available in a plasma membrane places an upper limit called the transport maximum on the rate at which facilitated diffusion can occur. Is that also existing for the channels? A maximum transporter, a maximum diffusion through membrane channel? Once all the transporters are occupied, the transport maximum is reached, and a further increase in the concentration gradient does not produce faster facilitated diffusion. As soon as it's completely full, it doesn't go faster, it doesn't change if there are 1,000 or 100,000 people waiting to go over. It is only possible to take 100 or whatever the number is every time over. Thus, the process of facilitated diffusion exhibits saturation. Solutes that move across plasma membranes by facilitated diffusion include glucose, urea, fructose, galactose, and some vitamins. Glucose enters many body cells by facilitated diffusion as follows, in figure 310. Glucose binds to a glucose transporter protein called GluT at the extracellular surface of the membrane. GluT undergoes a conformational change, and GluT releases glucose on the other side of the membrane. Is the thing actually going as we're showing it here? That way? It's just going open and close, open and close. That looks stiff here. I described the other ones here as a snake. We're taking it in and bringing it over. That one looks just as it comes in, and then it's, I don't know what the word for it is, it's just to the other side. When it's released, that would answer my question, how is it released? It's just released so that, and if it closes again, there happens to be one, and it goes over to the other side. Is that like, just like a, what is the name for it? Where ships go through or whatever. It just happened to be there in that area. It closes, and then it happens to go over through the other side. Is that a happen to be the situation, or is that something planned? After glucose enters a cell by facilitated diffusion, I'm wondering if it makes sense to call it diffusion. Well, now where we have it so picturesque, so illustrated. So, after glucose enters a cell by facilitated diffusion, an enzyme called a kinase attaches a phosphate group to produce a different molecule, namely glucose 6-phosphate. But that is not part of the facilitated diffusion, it is just part of how glucose works, I think. So glucose seems to work like if when the glucose is over, there's directly coming someone else and binds with it. This reaction keeps the intracellular concentration of glucose very low, so that the glucose concentration gradient always favors facilitated diffusion. I had already just 20 seconds ago the picture with someone eating it, with there's food coming in. And it makes now a lot of sense. If the breadcrumbs are all just around there, then next time that thing closes, it just takes free over and there again. Wait, if the dolphin or the fish wants to make sure, or the turtle, that they're staying there, they have to eat them, so that they're not accidentally being brought out again. And that is the way the glucose is directly bound, so that it's directly not able to go over again. So that always is the glucose coming. This reaction keeps the intercellular concentration of glucose very low, so that the glucose concentration gradient always favors facilitated diffusion of glucose into, not out of, cells. The selective permeability of the plasma membrane is often regulated to achieve homeostasis. For example, the hormone insulin promotes. I would at that place have another definition of what homeostasis is, or the way say what homeostasis is, because what we had in another book in biology that we did in another series in biology was that it's just to keep the human body at its state as it is. It's the running level, the temperature, the water content, the sugar content, everything. Just keep it at the running level. That a large part of that book seems to go over how it actually works, and that other book seems to just to tell that is the name for it and what is happening, and that book goes now every 50 pages for one of these things. Now I would like to have a new definition based on, for people wanting to read that book and not just the little definition. I wanted to know really what is homeostasis, now with an eye on that we're having over 1,000 pages explaining what is happening there exactly. So is my saying what I just said there oversimplified? Or is it that? Because what we're telling you that the cells are doing that's a little bit away. Maybe it is a part of it clearly, but what is now the new definition based on, if we not have one sentence to explain it or two, but let's say at least one page to explain what homeostasis is? As much as is necessary to explain it properly. For example, the hormone insulin promotes the insertion of many copies of a specific type of glucose transporter into a plasma membrane of certain cells. Thus, the effect of insulin is to increase the transport maximum for facilitated diffusion of glucose. With more transport is available, body cells can pick up glucose from the blood more rapidly. The hormone insulin promotes the insertion of many copies of a specific type of glucose transporter into the plasma membrane of certain cells. So insulin is the architect, the builder, the creator of, I don't know how exactly, of these glucose transporters. So insulin goes there and creates, however, a new transporter for glucose, and now more glucose can be taken out of blood into the body cells. That was the cytology part. If you are here for cytology, you can already take the next part of cytology, where we will continue with cytology. If you stay here and participate in the extensor carpi ulnaris strength training, then is that your arms. Good and still stand, arms up, hands hold the fists and bring them down and up. And that way around and again down and up. We're having 3 rules. The first rule is that we're stopping immediately as soon as there's pain. The second rule is that we're going only as far as we can go with a little bit of effort. And the third rule is that we're not overdoing it. We just want to strengthen, not to exhaust. You can use now a towel, a t-shirt or a sock, press onto it, down with the wrist, and up again. Press evenly with all 5 fingers. And again up. And the same with your right. Press and down. And again up. And with the other hand, press and to the outside. And in again. And on the other side, press and in. And with the other hand, press on top and down. And with the other hand again, down. And down. And just press. With the other hand, press. And It was the extent of carbohydrate strength training. I thank you very much and next time.